a while now. This is very, very exciting. Soil carbon's very exciting, James, I get that, but this is also very exciting. The boxthorn issue in the rangelands is a major issue, and it's not an easy issue to solve. But looking at it from a different perspective and looking at it from a fungus perspective is something that is truly exciting, and we're very lucky that we have sites in this area which Ben's going to be talking about, and we've been working with the CSIRO team out here now since 2022. I've made my first release in 2022 at the end of the La Nina, and we're just starting to see some really worthwhile results. But Ben is the expert, and please take the opportunity to talk to Ben and Caroline and Peter while they're here today, because it is really, and it's wonderful to have the CSIRO team here. So welcome, Ben.
Thank you very much. Sorry. Hello, everybody. It's very nice to be here. And thank you very much, Sally, for the invitation. My name is Ben Gooden from the CSIRO. As Sally said, we began working out here 2 or 3 years ago on this new project for African boxthorn, where we are trying to control the plant using a biological control agent. And many of the people in this room, many students, I haven't had the opportunity to speak to university students for several years when I used to be a lecturer based at the University of Wollongong before I started in this position. And I suspect that many of you in your courses may have touched on invasive plant management, weed control, but biological control agents as well. So I thought today for this talk I would introduce at the front end a bit of a primer for what we mean by biological control. Some people have different ideas of what biological control is and how it's used and the science underpinning it. So I'll first introduce that before we then talk about the African boxthorn itself. So we're very grateful to be working out here in this area. As Sally said, African boxthorn is one of the worst weeds which we have in Australia. It's within the top 20 of weeds of national significance. It's particularly problematic to control from a land use productivity point of view, but as well as for its environmental impacts to biodiversity, particularly along creek lines and riparian zones. And often in those more sensitive ecosystems, it is really problematic to control African boxthorn using chemical herbicides for 2 main reasons. The first is that the chemical herbicides themselves do have a biodiversity impact if not used correctly. And the boxthorn plants are highly resilient to herbicide control in many cases, so they can just recover over years from that. And for landholders, it is increasingly expensive to purchase chemical herbicides for use against African boxthorn. So the point of this project is to introduce a biological control agent, not to solve the problem for boxthorn, but to complement how people control the plant using chemical herbicide methods. Maybe before I move on, I will just give a bit more context about who I am and the CSIRO research that we do. The CSIRO is one of the few organisations working internationally that undertakes biological control for weeds. And the CSIRO have been doing that for over 100 years, with many successful programs, more than 270 weeds for which biocontrol agents have been released. The project that we have going on at the moment is one of about 20 different weed projects for African boxthorn using a rust fungus detected in South Africa for its release, and working with Peter and Caroline, who are here with me today. Following the releases, we are undertaking monitoring and evaluation to see how effective the program has been. Now, classical biological control first. We understand classical biological control to be where we introduce quote-unquote natural enemies of a target weed or pest, whether it be rabbits or some invertebrate examples here or weeds, where those natural enemies naturally occur and have co-evolved with the weed in question back in the native range of where that weed is from. In the case of African boxthorn, the weed is native to southern Africa, and the rust fungus that was collected and tested for release co-occurs with African boxthorn in South Africa as well. Classical biocontrol is about where we bring that naturally occurring organism into Australia, release it onto the target weed to calibrate the population of the weed back to a lower, more sustainable level. And it's based on this idea of the enemy release hypothesis. What we understand the case to be for many of these weed species is, when they come from their native range, as an example here for a weed, it might come from South America. It's introduced to Australia either accidentally or ornamentally or for some other purpose. It then has become naturalised and spreads through the environment and becomes invasive. One of the mechanisms for that invasion is that it no longer has the natural enemies with it to reduce reproductive output and growth. What we seek to do is reintroduce that, putting top-down pressure back on those weeds, and reduces the growth of the plant and its invasion potential. I'll just step across here and talk a bit more loudly. What we understand the case to be, this is weed density before a biocontrol agent is released. Weed populations always go up and down seasonally, or depending on what the level of rainfall might be in a particular year. Productivity of weeds is highly variable. A biocontrol agent is introduced at some point following testing. The population of a biocontrol agent rapidly expands, typically exponentially in the first few years to decades of its release. And then it begins this dynamic dance, antagonistic relationship between the agent itself and the weed. The populations are cyclical over time, but this dynamic equilibrium is reached again, where the weed population is much lower than what it was before the biocontrol agent was released. And one of the classical examples of this is skeleton weed. There are many different examples of weeds we work on, but skeleton weed was one of the most successful in a production sense, for which the CSIRO began research in the 1950s and 1960s in Europe. What this graph represents here for skeleton weed in the early 1970s, a biological control agent, a rust fungus, was detected in Europe. Research was done on that to ensure that it was safe for release in Australia. The fungus was released in the early 70s. And then after about a decade, the population of the weed collapsed to about 80 to 90% of what that original population density was. And the production of cropping systems and grazing systems in areas where the weed had an impact, that productivity increased as well. A couple of points I want to make about this is, following release of a biocontrol agent, if an agent can establish well, that's a good thing, but the benefits of that really accumulate over very long periods of time. We're talking at least multiple years of sustained impact over the decadal scale for those impacts to be borne out in perpetuity. Biological control agents that are released do not have immediate impacts. They have to establish first and widely in the landscape and have the ability to spread before we begin to see negative impacts, as we would like to, on the weed. But if it can be cracked, we're talking about multiple back-to-back years of accumulated benefits in perpetuity over the decades. So for Australia, more than 270 agents have been released, approved for release on more than 60 weeds. And a review undertaken on those programs a couple of years ago indicated that about 40% of those weeds targeted, there had been achievement of the desired benefits. That's either reducing the competitiveness of the weed, increased productivity in agricultural landscapes that are otherwise impacted by the weed. It also means reduced ability for the weed to spread. Typically what we see with biocontrol agents is, if a weed is heavily infested by an insect or a pathogen, its ability to reproduce is suppressed. For most of these weed species, their ability to spread very quickly across the landscape is contingent upon rapid reproductive output. So if we can break the back of reproduction, you can dramatically reduce the landscape scale invasion risk. And we typically have multiple phases to our research. The first, which can take many years to decades, is what we term native range exploration, where researchers work within the native range of where the weed is from. In Australia, we typically undertake exploratory research internationally in the United States, Central and South America, Southern Africa and Europe are the places where most of our research is undertaken. When we identify a candidate biocontrol agent that looks promising, we then import the biocontrol agent under permit to high security containment quarantine facilities in Australia. The CSIRO maintains 2 of those facilities, one in Canberra and one in Brisbane. And our research has taken, on average, it used to be on average about 6 to 10 years, but the efficiency by which we do the research has increased. So our research in a quarantine facility can take between 3 and 4 years now to determine that the agent is indeed safe and will not have any non-target or negative effects on any native or any economically important plant species in Australia. Once permission is given for its release, we then undertake a mass rearing and release program where we translate the agent from a lab into the field environment, and then we monitor the effectiveness of that. And this is the stage that we're at for the African Boxthorn project, which we started a couple of years ago here working with Sally in the Hay region. So some of the successful programs we have had recently, and we don't just work in a rangelands environment, we work in subtropical and temperate rainforests. For this weed here, which is wandering trad, we released a biocontrol agent, a fungus called Cordiana for that in 2020. After 5 years of sustained effect, we're getting significant reductions in the abundance of the weed. The further north you get in New South Wales, where it is warmer and more humid, the fungus tends to work better. And we're getting up to 90% reduction in the weed. And most importantly, in some areas where the weed has been reduced by that amount, many native plants are recovering in that ecosystem. We have projects also working with insects. Here is an aquatic weed called kabomba. It is a significant problem for irrigation infrastructure and water security, and a weevil has been identified for it in Southern America and given permission for release, and it's looking pretty good as well. And then we also do have some rangeland projects, but this one in particular for Hudson pear is being led by New South Wales DPI, where there is a cochineal insect released for it, and it has really rapid, significant negative effects on the weed, where the entire weed itself can collapse under the pressure of attack from the cochineal insect. So if we move on to African boxthorn now, we have a rust fungus within the genus Puccinia. It's called Puccinia rapipes, and it was detected in southern Africa in about 2015-2016. Now, African boxthorn, I'm not sure if many people here, the visiting students, are familiar with this plant, but certainly landholders in the area will have to battle this plant often and will be highly familiar with it. It is within the genus Lycium, species Ferrocyssamon. It is within the family Solanaceae, like tomatoes and the nightshades. It's native to South Africa and was brought to Australia about 150 years ago as an ornamental plant, but also for planting widely in the landscape as a hedging species. I didn't ask Caroline if I could put this picture in, I hope you don't mind, but it's one of my favourite pictures because in an area where it is unconstrained growth of this plant, the biomass accumulation is significant and the plants can be as large as the ones you see here. One of the main problems that we face for this plant in a rangeland context is, particularly in summertime, it accumulates around the base of old trees within paddocks in particular. It is a bird dispersed species, and so the fruit are carried in by birds who roost in the trees, and then the fruit are defecated and the seeds can germinate. But problematically, it reduces the capacity for those trees to function as shade trees and retreat trees for grazing livestock. As I said, it's a weed of national significance. It is prioritised for management across Southeast Australia. But increasingly, the feedback we're getting from people is, look, even if I can afford the chemical herbicides, and even if I do have the time for mechanical control, it's extremely costly. It's very difficult to control using these classical techniques over the size of properties that people have to manage. But what we're also seeing is we're starting to notice as well that many plants which are sprayed with chemical herbicides, there's significant capacity for those plants to regenerate following control of using chemical herbicides. So the rust fungus was brought in. It's native to South Africa. It's tightly co-evolved with African boxthorn. And we understand from our testing that it has no ability to complete its life cycle on any other plant species. It is an obligate biotroph that can only complete its life cycle on African boxthorn. What we also know about the fungus is that the spores are hardy and can spread long distances by wind. So this is the general life cycle of the fungus. What happens is the plant is infected by the fungus with microscopic spores, and we can't visually see that process as it's unfolding. It is a microscopic organism at this stage. When the spore germinates, a hyphae of the fungus will penetrate into the leaf surface. The interfacing with the leaf, it is penetrated by the fungal hyphae. The fungus infiltrates that leaf, and after about 2 weeks or so, Caroline, I think you can get this yellowish flecking, which is an indication that the leaf is infected. But most importantly, what happens is the fungus hasn't got the ability to grow its own food like a plant can. The plant will produce sugars for itself through photosynthesis, through fixing carbon dioxide. But what the fungus does is it takes a fraction of that carbohydrate that the plant would otherwise use for itself for growth. So in that sense, the fungus is parasitic. Through that effect, it depletes, it reduces the productivity of the plant itself. What we anticipate that would mean is there's less carbon available to the plant to reproduce, for instance. So it disrupts the carbon budget for the plant. Then what we see a couple of weeks after that, these darkish patches form on the surface of the leaf, which are what we term lesions of the fungus. And within each of these lesions is where the fungus reproduces again. And it completes its life cycle. Spores erupt from those lesions and can go on to infect a nearby plant dispersed by wind. And then shortly after that, when the fungus completes its life cycle, the leaf that's infected begins to die off. So the fungus is effective. It acts in a biological way to control the plant, but it does not act in the same way that a chemical herbicide would, which is systemic, and it can cause rapid death of the plant when sprayed with a chemical herbicide. The fungus itself may kill off the leaf, but it will not kill off the entire plant altogether. So we're at the point now where we are undertaking field releases of the fungus. The mission of our program is to release the fungus as widely as we can in New South Wales. One of the limiting factors in a successful biocontrol program is whether or not you can get the biocontrol agent established in the first place. If we can't get it established, then it hasn't got the ability to naturalise, it cannot spread, and more importantly, it won't be able to impact the weed populations as we would like them to. So for the first 2 to 3 years of the project, really our work is to engage with landholders, government, other community groups, and so on, to get the fungus established to as many different locations as we can. Following on from that, of course, we do need to monitor and evaluate the effectiveness of that work. Can the fungus establish, yes or no? Is the fungus able to persist across different seasons? We're especially interested to understand if you release the fungus in 1 year and it can establish, can it actually survive the summer and then re-establish in the population the following year again? Or do you need to supplement that through further releases? We don't quite know the answer to that yet. Importantly for us, we have set up our own experimental release plots where we can undertake detailed monitoring and evaluation over time. But all of this work is delivered in partnership with landholders, government and community. The way the fungus is released, I'm wondering, Caroline, if you can, I forgot to get the release kit, if you can bring that up. Thank you. Thank you very much. Caroline is the lead pathologist who works on this project, and the program went through a year and a half of optimisation to get to this point where you can send out a release kit to farmers and landholders and others. Essentially what we do in the laboratory is we culture viable spores of the fungus, which can be a tricky process, I think, Caroline, because you have to get the spores exactly right. If they're collected too early, they won't be viable. If they're collected too late, they won't be viable. Once they're collected, they have to be dried and stored in a particular way to maintain the viability in a form that can be packaged and sent to people. So that took a fair amount of time to optimise, and then we also had to optimise the recipe by which, when we give the fungus to people, there's a little recipe that people can follow to be able to release the fungus effectively. So and I can pass this bag around, we give people a small vial containing viable spores of the fungus. We give people a plastic bag, as you can see here, I'll explain in a moment, and we give people a spray bottle so that they can activate the fungus in the water, give that a bit of a mix. You need to include another chemical here as a surfactant to help with breaking up the fungal spores and ensuring that they stick to the leaf and that they don't run off with the water. And then bagging up the stems of the plant that are sprayed with the fungus, because it needs to be maintained a humid environment for at least 8 to 12 hours. The other catch is that the fungus is sensitive in the establishment phase to high temperatures and sunlight, which is a little bit tricky because we have to make sure the fungus is released as late into the day as possible just before sunset. Infection will occur overnight, but then the bags have to be removed as early as possible the next day before the sun gets too high to ensure that the fungal spores are not killed off the next morning. So if people can commit to the recipe that we give to them and invest a little bit of time in the bagging and the taking off of the bags, we are getting pretty successful rates of fungal establishment. So suitable sites for release we are finding is shaded woodlands are suitable. The plants grow very well under canopy, but most importantly, these canopy trees moderate the temperature of summer temperatures in particular, taking the edge off the fungus and allowing it the best opportunity for establishment. We are also identifying that creek lines and river embankments are especially good for the fungus because those environments, even in the height of summer, overnight maintain a really high local humidity. And what we know the fungus does once it's in the leaf, it doesn't require humidity. But when it's producing spores that have to go off into the environment to find another plant to infect, that's the step that is benefited by humidity. So local creek lines and river embankments are especially good. Releasing on cool, overcast days, we know, also improves infection. Releasing the fungus in autumn, winter or early spring is suitable, but releasing it any later in spring or early summer, we are not getting good levels of infection at that point. So we're very grateful to local land services who have partnered with us on delivery of this project throughout New South Wales. And today is just one example of the type of workshops that we run for this project. We have releases made throughout New South Wales in the core distribution of the weed, at least 70 release sites at the moment, with many more coming in autumn 2025, but the project does run through until June 2026. So there's still plenty of opportunities for people to register and participate in the program. So to that end, what I encourage you to today, either with us directly, please come and have a chat, or after the workshop, if there's still interest, you can contact Sally and Sally will pass on your information to us. We'll be here at morning tea. We have registration forms available. There's no obligation to participate, but if you wish to get more information to help you make a decision, register and then we get back in touch with you. So to finish off, I just want to share with you the type of experimental releases we're doing. the talk that you gave before, James, was excellent to give context around you can have all of this technology that you wish to test and trial in collaboration with community, but running in parallel to that, you need to collect good quality data to be able to demonstrate the efficacy of the type of technology you're working with. So we've set up, the CSIRO has 16 permanent fixed monitoring plots throughout New South Wales where we have experimentally released the fungus ourselves, where we are monitoring intensively every 6 months to determine, is the fungus establishing? If yes, can it then spread to nearby plants? If the answer to that is yes, are we actually seeing over time a reduction in the health of the plants? And if so, how quickly is that happening? When we released the fungus in November, December 2023, that was the first time we had the opportunity to do so, but we suspected at that time it was the wrong time of year. It was far too hot and very, very dry. When we went back in the May of 2024, we did not detect the fungus anywhere. We suspect within the week of us releasing, the temperature went up to high 30s, low 40s at the most vulnerable time for the fungus. So we weren't surprised that that did not work. We had the opportunity to release a second time in May of 2024 or 2023? 2024. We detected the fungus at every single site at which we released a second time in the autumn. And now what we're seeing at these sites, including in Dubbo in the Central West, in sites which are really exposed, they have canopy overhead, but they're not near any water supply. Forbes, and also down here along the river. When we came back, the fungus had become very well established at very high densities, and I purposefully took these photographs of the fungus on plants that we hadn't released it on. So the fungus had been released, completed its life cycle, and then went on to spread these nearby plants. So we're very pleased with this level of infection. Before I finish off, I just want to say that the trick with this fungus is, it is only at this point of reproduction when we can visually detect its presence. The rest of the time it is microscopic within the leaf. So there's a little bit of tree falling in the forest. If we turn up and we can't see it, does that mean the fungus is there or not? So we're now routinely collecting leaves, and we're starting a molecular diagnostics program using eDNA to determine whether or not the fungus is present within the leaf before we can actually see it. The leaves drop off very quickly after infection, so if we're not there to see the infection, the leaf might drop off. So the false negatives are a bit of an issue with our program, but we'll use DNA to cut through that. So to finish off, the likely outcomes I wish to share are that the really short-term impacts are not likely to occur for this fungus. It is a classical weed biocontrol program where the benefits are likely to accumulate over many successive years to decades. It is not a chemical herbicide. It cannot be used in lieu of or to replace the use of chemical herbicides. It also will not kill the host plant, but we anticipate it will reduce the reproductive output of the plant and the growth of the plant. So for many landholders who either have an obligation themselves through regulation or wish to very quickly improve the productivity of their landscape, if you need your plants removed altogether, the fungus can't be used for that purpose. But after you remove your plants, if the fungus is well established in the landscape, it would reduce the risk of the plant reinvading back into your property. So it's not a silver bullet, it's not a chemical herbicide, but at the landscape scale, we're aiming for this to be a complementary tool to those other classical methods of control. So thank you very much for the invitation. We're very pleased to be working with you, and thank you for your welcome this morning. It's very nice to be here. If we're here for morning tea, I'm at my time, so there might not be enough time for questions, but please come and have a chat with me and the team at morning tea if you want more information. Thank you. Thank you.
We will take questions.
Okay.
I can assure you we're definitely taking questions.
Okay.
This is a huge topic for our area, and we'll make up time. So landholders, while we've got the floor and we can share the information, let's have a go. I'm sure people have got questions. They'd have to have questions.
Hello. Hi, I'm Lucy from Sydney. I was just wondering if there's any chance that the plants could develop immunity to the fungus?
Again, the question was about whether the plants can build up an immunity to the fungus. The answer is yes, but over very long evolutionary timeframes. The fungus and the plant are co-evolved with each other, and through co-evolution, the plants evolve to be more resistant, whilst at the same time for the fungus to survive and maintain its evolutionary viability, it increases how well it is able to infect the plant. So the co-evolutionary dynamics means that they're really in tight arms race to be more resistant from the plant's point of view, but to be more highly infectious as the fungus. Over the last 100 or more years of any of these programs being undertaken, there's been no evidence for any of these weeds that they're developing increased resistance, likely because the fungus can keep up. It's evolutionary prerogative to do so, but that's a great question.
Hi, I'm Kimberly from New Zealand. With the fungus not being something that will kill the plant, what's the incentive for people to want to use it if they just have to use mechanical or chemical versions to control the plant anyway?
That is an amazing question, and it is something that we have been grappling with, that benefit, because as I showed with the earlier examples of weeds, some of those are reducing the abundance of the plant dramatically over quite a short period of time, and they have very clear benefits. This is a much more complicated situation. However, one of the feedback we routinely get from farmers is, even if they mechanically control around the base of a tree to increase access to shade trees for stock, over subsequent years, the weed rapidly comes back in because of bird-dispersed fruit, often coming in areas where landholders can't access to control. So it could be creek lines managed by council, or creek lines in conservation reserves, like we work at a national park at Barubin. If those plants there are not controlled, the spillover effect will continue, and then the burden will be on the farmer in perpetuity to maintain the weed reinfestation on their property. So what we're actually encouraging people to do is, if on your property you have the weed that you need to get rid of, don't release the fungus on it. Find complementary areas in the landscape. It could be tree lines or fence lines that you share with a neighbour, in areas where you may not have capacity or time to control the weed anyway. Just get the fungus established in the landscape. So it's complementary, but it will, and it's a good point, it means the desirability for the fungus may be lower than otherwise, but we just need to find what the complementary point is and then target that.
My name is Isabel from the University of Sydney. So just to cover again, sorry. So first of all, how does the rust fungus interact with native fungus species, and do you have any concerns about its evolution, given the short research period you guys have, in evolving to affect native species or interact further with native fungus and plant species?
Great, okay, excellent. The fungus does not interact with other fungal species. The mechanism of reproduction means that it doesn't cross with other fungi. It is a selfing fungi, and so it will self its own spores asexually. So that's a really good point, because when undertaking a risk analysis for a project like this, you have to understand the biology of the organism. If the fungus was able to reproduce with other fungi, and therefore through recombination and hybridization have new combinations of genes, this fungus would never have been considered at all for release. So that's a really astute, intelligent question to ask, because it doesn't come up in conversation often. In terms of its potential negative effect to native vegetation, this type of rust fungus is one of very few organisms which is an obligate biotrophic fungus. The spores have no ability to complete its life cycle if it interacts with any plant other than boxthorn. If the spores land on the surface of another plant, it interacts chemically with the surface of that leaf. If it's chemically through signalling mechanisms, if it's not compatible, it won't germinate at all. So that formed the basis of all the risk analysis for 10 years, is to see if the fungus can infect anything else, and if it can't, that lends itself to being safe for release.
Me again, sorry. With releasing the fungus on the plant, does it disturb the reproduction from the bird's seed dispersal? So if it has it and the birds eat it, will they still be able to spread the plant?
Okay. What it will do is just reduce how many seeds the plant can set. When a seed is produced, it tends to be viable. So it is just about reducing how much fruit can be produced at any one time. There are only a few bird species in this region which would eat the fruit anyway. And in fact, one of the main fruit dispersers for boxthorn in the rangelands environment are foxes. So there are these complementary and dual benefits as well. We also know that boxthorn is highly attractive as for rabbit colonies, warrens as well. So, but what concerns me is the rate of time over which fruit depletes. We're talking about years to decades of benefits.
Wow.
Thank you.
Thanks, Ben. Thank you so much.
Thanks for the invitation.
No, that's fine. This is a huge topic for our area, and it's fantastic that we've got Ben and Caroline and Peter here today.